Hello students, welcome back to the channel. In this video, we are going to give you a quick revision of this chapter. I hope this video will be helpful for you. Now let us start. So now we are going to start with the centrosome. The centrosome is an organelle containing two cylindrical structures. They can ask you with respect to the shape. And two cylindrical structure that is known as centriole. Now see the diagram carefully. So first of all, this pair of cylindrical structure, pair is known as diplosome. And this cytoplasm which is present around it. This is a cytoplasm which is present around it. So cytoplasm around it is known as centrosphere. That is why this organelle name was centrosome. This much is clear. So this is, if you see this one centriole, you're finding this one centriole is actually having 9 plus 0 arrangement. 9 plus 0 arrangement. 9 is what? 9 are microtubules. 9 microtubules in the form they are present in the triplet form. So this is your 1, 2, 3, 4, 5, 6, 7, 8, 9. And this is your triplet. So 9 peripheral microtubules in the form of triplets, they are present. And in the center, nothing is there. And these triplets are interconnected with the help of the linker. This is C2A linker. Why I'm saying C2A? Because this is A, B, C. So A, B, C. A is always inside. C is outside. So this linker is known as C2A linker. So this is C2A linker. Okay, so this is C2A linker is there. Now from the A, radial spokes will come. These are the radial spokes which will join in the center proteinaceous hub. So radial spokes are joining in the center, that is known as proteinaceous hub. That is why this arrangement is known as 9 plus 0 arrangement. This arrangement is known as 9 plus 0 arrangement. Is it clear? Now let us see the NCERT. So they are surrounded by amorphous pericentriolar material. Both the centrioles are perpendicular to each other. That is why in the diagram I was drawing this way, because they both are perpendicular to each other. In which each is having organization like a cartwheel. They are made up of 9 evenly placed peripheral fibril of tubulin protein. And tubulin protein are the one which is making the cytoskeleton microtubule. Please remember, each of the peripheral fibril is triplet, and they are linked also, adjacent are linked by C2A linker. And the center part of this is known as a proteinaceous hub, which is connected with the tubule of peripheral triplet by the radial spokes. Is it clear? So I think so this much is clear to you. Now centrioles, they form the basal body of cilia and flagella and spindle fiber to give rise to spindle apparatus. Now wait with it. Now see cilia, flagella, this is your cell. And this is your microtubule present. Now analyze the diagram very carefully. What will happen? I'll use this orange color only. It will go up to form the exoning. So this protruding out part, this is known as exoning. And what is this part? This is known as basal body. So who is making the basal body? Centriole only. If you remember, I have drawn these two. It means centriole is the one which is making the basal body, and that is only the one which is going up to form the exonym of cilia and flagella. Now, when we see the exonym of cilia and flagella, what we are finding? There are also 9, peripheral 9, but this peripheral 9 will be doublet. Wait, 3, 4, 5, 6, 7, 8, 9. 9 peripheral doublet. And remember, it is A and B. C is not in the diagram, A and B. Now from the A, make two dynenams. From the A, makes two dynenams. And connect them. This is from the inner one to the B. Inner one to the B, connect this way. Clear? And in the center, you are finding central sheet where two singlet are present. So we will call this arrangement, which I'm drawing of exoning, we will call this arrangement as 9+2 arrangement, but this 9 periphery is doublet. And now we have to draw the radial spokes. So radial spokes will come from A only, and it will be this way. Radial spokes. Touching the central sheet. Is it clear? So we say this arrangement is 9+2. Another diagram, just to make you learn only, do one thing. I'll just give you imagination, 1 minute. This is your cell membrane, which is going up to form the exoning. Clear? This is your A microtubule, which is going up from the basal body to upside. This is your B microtubule, which is going from the basal body, means from centriole to upside. This is B microtubule. Clear? Then this is your C, which is not able to go up. Learn this way, it will be helpful for you. So this is A, B, C, A, B, C. Clear? And now you do one thing in the center for your learning. Draw this way. Central 2. And this is your radial spokes coming from A, which are covering the, which are touching the central sheet. Just to make you learn only, this is the way if you learn it. So you'll get an idea. If centriole is there, it means cilia flagella can be made. So in case of eukaryote, centriole is the one which is giving rise to the basal body of cilia and flagella. If basal body is there, then obviously exoning will come. Clear? And here, make it from the, imagine this is one line, which is not central proteinaceous sheet. And the radial spokes are touching it. This way, if you will learn it, I think so it will be helpful for you. So in the basal body, what is your, you will write it. In the basal body, you'll remember 9+0, and this 9 is triplet. And here you are saying exoning, and exoning means here you are saying 9+2, and this 9 is doublet. So cilia and flagella, they are hair-like outgrowth of the cell membrane. Cilia are small, but work like an oar. Okay, they are the one which are working like an oar, causing the movement of either the cell or the surrounding fluid. Flagella are long, and they are responsible for cell movement. So please learn it. Cilia can make the surrounding fluid also move, but flagella is responsible for cell movement only. Prokaryotic bacteria also possess flagella, and we studied yesterday that prokaryotic flagella is having 3 parts: basal body, hook, and filament. Remember, this filament was unistranded, and it is made up of which protein? Flagellin protein. So this is in case of prokaryote. Now for eukaryote, what you have to remember? Eukaryote, if you see, the electron microscope studies of this shows that they are covered with the plasma membrane. I think so my diagram is helping you to learn it. Their core called exoning is having a microtubules, running parallel to the long axis. Exonym usually has 9 doublet microtubules and a pair of centrally located microtubules. So you can write here basal body, because right now we studied centriole will make basal body, and exonym. Exonym is having 9 plus 2 and basal body is having 9 plus 0. And this arrangement of the exonym, we call it as 9 plus 2 arrangement. Central tubules are connected by the bridge and are also enclosed by the central sheet. And they are connected to one of the peripheral tubulet also by the radial spokes. So both the cilia and flagella, they emerge from the centriole-like structure called basal body. Clear, beta? So you can see here, this is the plasma membrane. Peripheral microtubules are doublet. Here interdoublet bridge is there. Okay, and these are your dynamics, the one which is they have drawn it. This is a central microtubule 2 and which is surrounded by central sheet. Radial spokes, they are not touching the central sheet. Please remember in this diagram. So if you have learned the centrosome, obviously cilia and flagella is easy for you to remember. 3 types of cytoskeletons are there. One is microtubule, one is microfilament, and third is your intermediate filament. Right now we studied that microtubules are the one which are making centrioles. Means tubulin protein. Tubulin protein is the one which is making centriole. It means if the cytoskeleton is there, then centrosomes, they can make it. If centrosomes they are able to make, it means they are able to make basal body. And if basal body is there, cilia flagella will be there. So can I say microtubules are responsible for ciliary and flagellary locomotion? So they help in the motility. Microfilament. Filament, it is actually made up of actin protein, mainly actin protein. And you know, actin protein is contractile protein. And it is responsible for amoeboid locomotion. Microfilament are responsible for amoeboid locomotion. So we see in the locomotion chapter, cytoskeleton elements like microfilaments, they are helping in amoeboid movement. And amoeboid movement means with the help of pseudopodia, and pseudopodia means role of the cell membrane. So see here, that is why we say they are helping in maintaining the shape of the cell. And obviously when we say skeleton, so their work is to give mechanical support. Is it clear? So 3 cytoskeleton, please remember: microtubule, microfilament, intermediate filament. Just go in the detail here. See diagram, I'll show you. See better, these are the 3 types. Microtubules, they are hollow, diameter 25 nanometer, 15 nanometer in this way, made up of tubulin protein. Actin filament is making microfilaments, and intermediate filaments are made up of keratin protein. Microtubule is responsible for ciliary and flagellary locomotion. Microfilament is responsible for cytoplasmic streaming and for amoeboid movement. And intermediate filament is responsible to maintain the organelles in their position. Organelles should not move. They are responsible for that. You know very well microtubule. Right now we studied that they are the one who are making the spindle apparatus. Just 1 minute. Beta, if you remember, we studied right now the centrioles will make the help in the separation of the sister chromatid. Means helping in making the spindle apparatus. And you know, spindle apparatus are made by the assembly of microtubule. It means microtubules can go for assembly and disassembly. And in the locomotion chapter, we have studied that this is actin. Actin is made up of, each actin is made up of G-actin. Each actin is made up of G-actin. It's a polymer and it is made up of G-actin. It means microtubules and microfilaments can go for polymerization and they can go for depolymerization. But in case of the intermediate filament, they will not go for such type of things. There will be no polarization, no polymerization, and no depolymerization. So I think so cytoskeleton topic is clear to you, beta, huh? Now let us see about the next organelle, ribosomes. Ribosomes we studied only. The ribosomes are the one which are protein factory. And what are ribosomes made up of? Ribosomes are made up of ribosomal RNA plus protein. Now who has observed them? It was observed by Palade. That is why they are also known as Palade granules. Please remember this question, they are asking it. That is why they are known as Palade granules. When they are made up of ribosomal RNA and protein, so we call them as they are ribonucleoprotein. They are ribonucleoprotein. And this is a membraneless organelle. This is the organelle which is present inside mitochondria and chloroplast also. So we say one question can be framed this way: organelle present in organelle. Answer will be ribosome, because our ribosome is itself is organelle, and it will be seen not only in the cytoplasm of eukaryote, it will be found in mitochondria and chloroplast also of the eukaryote. So this is an organelle which is present in another organelle. Now, in case of eukaryote, the eukaryote ribosome is of 80S and prokaryote ribosome is of 70S. Is it clear? So you can do one thing, may write this way. 80S is divided into two. It is having two subunits, 60S and 40S. Same way, 70S is having two subunits, that is 50S and 30S. So it is given this only. Each ribosome is having two subunits, large and small. The 80S subunits are 60 and 40, and 70S subunit is 50 and 30. Okay, but I don't do the math work here. No need of adding and saying कि 60 plus 40 will become 100. No. Individually, they are coming this weight. Now, what does S stand for? S means Sedgwick unit, which stands for sedimentation coefficient. It is indirectly. Please, now here they can change the question and they can ask you, it is a directly measure. No, it is indirectly a measure of density and size. So I think so ribosome also. And see one thing, two types of ribosomes are there. Not only on the size, depending upon where are they found. If they are present free in the cytoplasm, we say free ribosome. Or if they are bound, just like on ER we studied in the previous class, then we say bound ribosome. Bound ribosome or free ribosome, every ribosome will make protein. But bound ribosome will make protein for secretion. That is why we are saying Golgi bodies are doing packing and helping in secretion. Secretion means they are releasing the things outside the cell. And free ribosomes will make protein for the cell purpose. It will make for the cell purpose. So I think so this much is clear, beta. Now let us start with the two cell organelle, that is mitochondria and chloroplast. You know very well it is a double membrane organelle, outer membrane. Inner membrane is finger-like projections known as cristae. So this is outer membrane. This is inner membrane. And this finger-like structure, they are known as cristae. And this space is known as matrix. And this is your intermembrane space where the proton will be accumulated to create a proton gradient. And proton gradient is necessary because that gradient breakdown will only help in the ATP formation. And for making ATP, you want the help of an enzyme ATP synthase, which is present on the F0F1 particle. This is your F0F1 particle, also known as elementary particle, also known as oxysome. Number will be more on the cristae. Okay, so these are your F0F1 particle, also known as elementary particle, also known as oxysome. You are finding here it is having their own DNA. We call it as mitochondria DNA. And it is having ribosome 70S. If you check this diagram, you will feel, if I don't show you the mitochondria word, you will feel I've drawn as if a prokaryotic cell. The mitochondria is actually living inside the eukaryotic cell as an endosymbiont relationship. So according to this origin, how these organelles are originating, according to their theory, they believe mitochondria and chloroplast are prokaryotes sitting inside the eukaryotic cell. As in which relationship? Endosymbiont. The mitochondria is actually gram negative aerobic bacteria. We believe mitochondria is gram negative aerobic bacteria. Why? Why I'm saying it this? Because if you remember in the transport chapter we studied, the outer membrane is having porine protein. And porine proteins are present in certain bacteria, in mitochondria, and in the plastids. If you remember in the transport chapter, open and check it better. Porine proteins, we say it is present in the mitochondria, chloroplast outer membrane, and in certain bacteria. And porine proteins are present in which bacteria? Gram-negative bacteria. And second important point is this mitochondria DNA is always showing uniparental inheritance. Because in the reproduction chapter we studied, it is the egg cytoplasm which we inherit. Sperm is only head is fusing with the egg, and head means only nucleus is fusing. It means the sperm cytoplasm is not entering inside it. So only fusion of the nuclei of the male and female occur, and the cytoplasm we inherit entirely from the mother. So we say mitochondrial DNA is maternal inheritance. This is only maternal inheritance. It is uniparental, only mother can give us. So it is the one which we are using to trace the evolution of the human evolution. We are studying because of the mitochondrial DNA, because that follow uniparental inheritance. Is it clear? Now, if you see 70 as ribosome, if you see DNA, so DNA is circular, only single circular DNA. So isn't it prokaryote? And if I'm saying it is prokaryote, it means it can reproduce by binary fission. Now let us see the NCERT. The mitochondria, unless specifically stained, are not easily visible under microscope. The number of mitochondria per cell is changing depending upon the activity. It means if the cell is more metabolically active, it will be having more mitochondria, because we see mitochondria as a powerhouse of a cell. In terms of shape and size, also changes are observed. Typically, it is sausage shape or cylindrical. Please learn all these things, all these statement questions, they can frame it. Okay, you should not lose marks here. Diameter 0.2 to 1 μm in average, 0.5 they will say, and length is this much. Each mitochondria is double membrane with the outer membrane and the inner membrane, dividing its lumen into two aqueous compartments, outer compartment and inner compartment. And inner compartment is known as matrix. So see here, beta, this is your outer membrane. Please learn the labeling. They are asking you this also. This is inner membrane. Inner membrane is thrown into finger-like projections, and these finger-like projections, which is known as cristae. And this is your intermembrane space, also known as outer compartment. Intermembrane space is known as outer compartment, and matrix is known as inner compartment. And matrix is actually a dense homogeneous substance. The outer membrane is continuous, but inner membrane is thrown into finger-like projection known as cristae. Now, beta, we have studied the word cristae here. And yesterday in the Golgi, we were studying word cisternae. So generally, when the students see the option, they get confused. Please remember, this is mitochondria cristae, and this is cisternae we study in the ER and Golgi. Clear, beta? The cristae increase the surface area. The two membranes have their own specific enzyme associated with the mitochondria. They are the site for aerobic respiration. It means they will produce a lot of ATP. That is why we call them as powerhouse of a cell. The matrix also contains single circular DNA, just like prokaryotes, single circular DNA and few RNA and ribosomes, which 170 is. And it means ribosome is there, DNA is there, so they can make their RNA. They can make few protein on their own. That is why we call them as semi-autonomous organelle. And I told you this feature resembles prokaryote. So we believe that they are endosymbiont, and when we believe that, and we are finding why do we believe it more? Because they divide by binary fission. They divide by fission method. Very important question. Is it clear, beta? Now see the plastids. Now first let me draw the diagram of plastid. See the comparison. It is also double membrane, and both membrane are continuous. There is no finger-like projections here. There is third envelope system. These are thylakoids. And in the thylakoid, the empty space, this space is known as lumen. This is lumen. Thylakoid lumen, we call it as. So this is stack of thylakoid is known as grana. So stack of thylakoid is known as grana. This is one grana. We can make another grana. This is the way granas are present. And between the two granas, you are finding stroma lamella. This is the stroma lamella present. Okay. Then apart from this, what you are finding, it is having a, this is what? Chloroplast DNA. It is having 70S ribosome. Clear, beta? And this is space. This is space which is in this area. This area which is left. This space is known as stroma. Please remember, beta, here we call matrix, here we call stroma. Stroma is a place where the dark reaction will occur. Grana is the place where light reaction will occur. Now these plastids, if you see evolutionary, they have started from the euglena, euglenoids, protista, kingdom protista. And if you see, we have studied in the kingdom plantae, green algae is having different types of chloroplast, cup shape, ribbon shape, spiral shape. Okay, so different types of shapes of the chloroplast are present. Sometimes some cell will have only one chloroplast, but mesophyll cell, in the case of the higher plants, mesophyll cells, one cell can have 20 to 40 chloroplasts per cell. So plastids, if you see, they all seen in plant cells as well as in euglenoids. Now, depending upon the pigment, they are classified as chloroplast, chromoplast, and leucoplast. Chloroplasts are the one which will have the chlorophyll and carotenoids. Is it clear? And chromoplast will have carotenoids. But both chlorophyll and carotenoids are lipid soluble. So chloroplast contain chlorophyll and carotenoids, which are responsible for trap the light. Chromoplast contain carotenoid, which is fat soluble, and carotenoid, two types are there, carotene and xanthophyll. Leucoplast is colorless. Now, leucoplast is of three types. But if you remember in the mouth, salivary amylase. Remember, beta, leucoplast, if you remember, beta, we have studied that salivary amylase act on the starch. So if they are storing starch, you call them as amyloplast. If they are storing oil, we say elaioplast. How to learn? See, beta, O-I-L, oil. Oil means lipid. So elaioplast store oil. And if you remember in the monocot seed, aileron layer is proteinaceous. So if they store protein, you say aileroplast. Whereas the aileroplast will store protein. So three types of plastid, depending upon the pigment is there or not. If it is there, then which color? And if it is not there, then what are they storing? Clear? So now, they are found in the mesophyll cells. And how many they are present? Please learn it, 20 to 40 per cell in mesophyll. Chlamydomonas is how many? It is Chlamydomonas is having only one per cell of Chlamydomonas. Chlamydomonas we study in the green algae. We have already studied they are of different shape. They can be ribbon shape, they can be cup shape, they can be lens shape also, oval, spherical, discoid. Please learn the length and the width also. They are the also one which is double membrane. Inner chloroplast membrane is less permeable. Please learn it. Inner membrane is less permeable because outer membrane is having porine protein. I focused it also while explaining in mitochondria. Space limited by the inner membrane is called as a stroma. And inside this, you are finding it, thylakoids are present. Thylakoids are arranged in the stack of the piles. This way, just like piles of coins to form grana. They are making grana. In addition, there is flat membranous tubule called stroma lamella. Stroma lamella is the one which connects the two thylakoids of the different grana. The membrane of thylakoid enclosing a space known as lumen, thylakoid lumen. The stroma contain enzyme required for the synthesis of carbohydrate. You will remember carbohydrate very easily because Calvin cycle is performed, photosynthesis. But beta protein is also, they are having the enzyme to make proteins. Because every cell will have the enzyme to make protein, because when they are having DNA, they are having the ribosome, obviously they are able to make their own protein. So this is again question area. Again, question area, beta. Stroma contain enzyme required for synthesis of, they will frame it carbohydrate only, protein only, carbohydrate and protein only, or carbohydrate, protein and fat. Then answer will go carbohydrate and protein. It contains small double-stranded circular DNA and ribosomes. Ribosome is 70S. Is it clear? So ribosome is also 70S. So we believe that they are nothing. They are aerobic bacteria. And they are cyanobacteria. Chloroplasts are believed to be cyanobacteria because cyanobacteria is the one who first developed the chlorophyll A. Is it clear, beta? Up till here, if you see, all the topics are covered. Check carefully, you will find your cell topic is covered, beta. Only last microbodies are left. So you have to just remember that they are minute vesicles which contain various enzymes found in both plants and animal cells. So from my side, the revision of this cell unit is over. You just check it. If you are having any doubt, then ask me.